Hello students, today we are going to continue with higher level topic 10.2. We're going to be looking at inheritance, and more specifically we're going to be looking at chi-square tests. So you might have done some chi-square analysis when you conducted your science fair experiment. Remember that this is a statistical test that is done to determine if you find any difference between what you observed and an expected frequency distribution. So this comes really in handy when you're looking at Mendel's experiments, because if, let's say, you try to reproduce Mendel's experiments, you pretty much know what phenotypic ratios he observed, and you want to then run your own experiments, and you want to compare what you observe versus what Mendel predicted to be the phenotypic ratio, let's say. So the observed frequency is what you observe in your experiment, and the expected frequency comes from a theoretical value. So in this case is what Mendel or other scientists, it could be even Morgan, have looked at and have predicted to be their expected frequency. So let's first quickly review what the difference between linked and unlinked genes are. So we studied unlinked genes, and this is what we refer to as Mendelian genetics. So these are genes that have equal possibility of being inherited, and you can get different types of potential phenotypic combinations. And this is due to the fact that the alleles can be independently assorted. And we know that this can happen because the alleles are found on different chromosomes. Okay. We've studied linked genes in the previous videos. And we know that linked genes will only express the phenotypic combinations that you find from the parents. So most of the combinations will come from the parents unless some type of crossing over occurs, right? And this is due to perhaps the fact that the genes might be separated by a larger distance per se, okay? So in linked genes, we know that the alleles are found on the same chromosome. So this is a review, right, of unlinked versus linked genes. Here we see the alleles found on different chromosomes from that parental generation. And for linked genes, we see the alleles found on the same chromosome in that parental generation, right? So when we're studying unlinked genes, we apply the law of independent assortment, which is Mendel's law. And in this law, we say that these alleles have a chance to segregate independently of one another. And this is where we do the FOIL method, and we independently assort these alleles, right? So first, outer, inner, last. And let me just quickly here do one for you. So this is what the phenotype looks like. So if we apply the FOIL method, we go first, right? Which is big B and VG for vestigial. So that would be this one first. And then we do outer. So it would be this one. And that's this one right here. And then we do inner. And that would be, where is it? Where is it? Oh, sorry. Right here. And then last. And then that would be this one right here. So we do the FOIL method to see how these alleles independently segregate from one another to form these four different gametes. And then we're crossing this parent with this parent, and luckily this parent is all homozygous, so we know that there's only one type of possible allele combination, right? So when we run the test cross between these four possible gamete combinations and this one, we learn after running the dihybrid cross that the parents, notice, can sometimes appear in the offspring, right? Here we see wild type and here we see black vestigial, but we also see different combinations like gray vestigial or black normal. So this is what an unlinked test cross is going to look like, and this is a Mendelian type of test cross. Now for this one, since it's on the same chromosome, right, we expect these two alleles to go together into the gamete, and here it is. And then we expect these two alleles to go together into the gamete, and there it is. And then we expect this parental allele to also go together into the gametes, and there it is, right, because the alleles are on the same chromosome. So notice that the majority of the offspring are going to resemble the parents, which is wild type, like the parent here, and black vestigial, like the parent here. Now, there's always a chance for recombinants, and this we studied also in the previous video. So there's always a chance for recombinants, but remember that the chance for recombination or crossing over depends on the distance, right, of these alleles on the same chromosome. So if you happen to have a large distance between these two alleles and a large distance between these two alleles, there is a greater chance for them to recombine, right? This one with this one, and then this one with this one, because they happen to be at a larger distance, separated by the centromere. So there is a possibility for recombinants, but notice that the possibility for that to happen is much less. So there's going to be much less recombinants than there are going to be phenotypes that look like the parents. So this is a linked test cross, and we learned earlier that Thomas Hunt Morgan was able to prove the existence of these linked genes in his theory of chromosomal inheritance, which Mendel did not know at the time. So this is just a quick recap, and let's continue. So chi-square tests. Your skill is to be able to use a chi-square test on data from a dihybrid cross. So you need to be able to fill out a chi-square test. So why would we do a chi-square test? So we try to measure a statistical significance to see if there's a difference between the observed and the expected frequency. Okay? If observed frequencies do not conform to those expected, then we say that we are dealing with an unlinked dihybrid cross. Okay? Because we know that either genes are linked and hence not independently assort, and that the inheritance of the traits are not random but are potentially being affected by natural selection. So let's start working on an actual example. So here you go through the steps of how to conduct a chi-square analysis, and there's 5 simple steps here that you should do. So you want to identify a hypothesis, a null versus an alternative hypothesis. You want to construct a table of frequencies, which is called a contingency table. You apply the chi-square formula, which you don't have to memorize. You determine the degrees of freedom, and you identify the p-value. And if there is statistical significance, the p-value should be less than the designated 0.05 value. So let's apply the chi-square application. So let's look at a simple dihybrid cross where you're crossing 2 heterozygous pea plants. So they're heterozygous for color and for shape. So we have big R is dominant. Big R stands for smooth peas is dominant over wrinkled peas. And yellow, big Y, which is yellow, is dominant over little y, which is green. So we know that Mendel observed these frequencies, right? And remember that I told you that anytime you have two heterozygous being crossed here, and it is an unlinked cross, the law of independent assortment happens, you're always going to end up with a 9:3 to 3:1 ratio. That's going to be always, okay? So when Mendel conducted these experiments, and remember that he conducted these experiments in his pea plants, and something to note here, and that we're going to look later, is that the pea plants that he used are just one variety of pea plants. There's different varieties of pea plants that contain different genes. But he used the Pisum sativum plants, and this is what he observed, okay? So if we want to run our own experiments and compare Mendel's expected phenotype, which is 9:3 to 3:1, and compare it to what we observe, then we do a chi-square analysis. So just like the steps said, well, first you're going to identify your hypotheses. And let's remember what a null hypothesis is. That means that there's no significant difference between observed and expected. And the alternative hypothesis is that there is a significant difference. So if there is a significant difference, we are going to assume that the genes are linked. And if there is no significant difference, we are going to assume that the genes are unlinked. Okay? So how do we create a table of frequencies? So we have our 1, 2, 3, 4 different types of possibilities that Mendel observed. The smooth yellow, the smooth green, the wrinkled yellow, and the wrinkled green. And let's say that he explored 1,260 total plants, right? Total pea plants. Now, this is what Mendel observed. And, this is what Mendel, This is what Mendel observed and predicted, okay? So this is Mendel's expected ratio of 9:3 to 3:1. So then when we run our experiment, we want to see if our observed results compare to Mendel's expected ratio, right? So we should get something like what Mendel got. We should also get a 9:3 to 3:1 ratio, right? So let's say we record what we actually observe in our experiment. Here, 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 and here. And let's say we run the experiment with the same number of pea plants that Mendel did. Okay. So next we have to apply our chi-square formula, which is this, where this symbol, sigma, is the sum, the observed frequency, and the expected frequency. And then we run some calculations of this formula. So this is what the Mendel expected ratio is supposed to be. Okay. So in other words, if we have a total of 1,260 plants, I observed to see a 9:3 to 3:1 ratio. And this is what I actually observed in my experiment. Okay, this, and this. So then I run some calculations so I can get my, plug it into my formula. So observed minus expected is 701 minus 684 gives me 17. Observed minus expected, 204 minus 228 gives me negative 24. Observed minus expected, 243 minus 228 gives me 15. And observed minus expected gives me negative 8. Okay. Then I square that and I divide by the expected value and I get these numbers. Okay, so that's the numbers that I get after I conduct all my calculations. So then what do? Well, let me move this up a little bit still. So then remember that the chi-square value is the sum of everything. So I have to add all of my values here. And I get a chi-square value of 4.76. Okay? So now what do I do with that value of 4.76? Well, I have to determine my degrees of freedom. And let's read here quickly what that is. In order to determine if the chi-square value is statistically significant, a degree of freedom must be first identified. The degree of freedom is a mathematical restriction that designates what range of values fall within each significant level. And this is the formula. Okay? But just to make it a lot simple for you, for a dihybrid cross, the degree of freedom should be the number of phenotypes minus 1. So in this particular case, if you look up here, you have 1, 2, 3, 4 possible phenotypes. So it's always going to be the number of phenotypes minus 1. So then it's 4 minus 1, and we end up with 3 degrees of freedom. So let's go down here. So there it is, 3 degrees of freedom. So after you have your chi-square value, which we just calculated up here, 4.76, and now you have your degrees of freedom, which is 3, now you can identify the p-value based on this distribution table. And this distribution table is given to you. Okay? So a value is considered significant if there is less than 5% probability, or p is less than 0.05, that the results are attributable to chance. So then what we do is we find our degrees of freedom, which is 3, and then we find our value where we're going to consider a 5% probability that the results are attributable to chance. So we're going to look at this value here, right? And we are going to focus on any number that falls below 7.85 is not statistically significant. Not statistically significant. And any number that falls greater than 7.815 is statistically significant. So in our calculation, we got a chi-square value of 4.76. So 4.76, right? If we look at this distribution right here, 4.76 is less than our p-value for a probability of 0.05. So what can we say? Are my results statistically significant, or are they not statistically significant? So 4.76 is not statistically significant, right? Because 4.76, it falls under my statistically significant value here. So what does that mean? What does that mean? That means that the difference between my observed and my expected frequencies are not statistically significant. Therefore, they are not different. They are not different. They are pretty much the same. So what that means is if I look at my observed values, they are very similar to Mendel's expected values. So if my results are not statistically significant, okay, that means that what I observed in my experiment was not different from what Mendel expected in his experiments. Okay, so pretty much what I'm saying here is I got the same results as Mendel. Okay, so in this particular experiment, if I get the same results as Mendel, then I can say that in this particular case, there is no significant difference between observed and expected frequencies, and my genes are unlinked. So that means that what the experiment that I conducted, the dihybrid cross that I conducted, is similar to what Mendel conducted, right? And remember that Mendel looked at unlinked genes. Okay? Now, in your activity that you guys are going to be working on later today, you are going to be looking at the discovery of linked genes, Bateson, Saunders, and Reginald Punnett. So the work that they did. And they discovered linked genes, but they weren't really able to prove it. Okay? So remember that Mendel's experiments were published in 1865, right? So when Bateson, Saunders, and Punnett, when they conducted their experiments in 1905, they were expecting to see the same results as Mendel. Now, here's the difference, though. They worked with sweet pea plants, and the genus species was Lathyrus odoratus plant, instead of Mendel's pea plants, which were the Pisum sativum plants. Okay, so they were working with the different pea plants, but they were expecting to see what Mendel saw. And this is what led them to scratch their head and be like, wait a minute, something's going on here, right? So you are going to read here in your activity, this up a little bit, that they started the same way as Mendel, right? But they were looking at other types of characteristics. So they were looking at flower color in their pea plant, but they were also looking at pollen shape, which is not a characteristic that Mendel looked at. And they crossed the parents, and then when they got their F1 generation and it was all heterozygous, they were like, okay, it's going well. And then they were expecting that when they crossed the heterozygous with themselves, so 2F1 generation, they expected to see the 9:3 to 3:1 ratio that Mendel saw as well. Okay, but to their surprise, that's not what they saw. And this is why a chi-square analysis is helpful, right? Because in this chi-square analysis, they were expecting to see this ratio, 9:3 to 3:1. So they were expecting to see these numbers here. So these are Mendel's expected ratios. But Bateson et al. In 1905, this is what they saw. This is what they observed. And it's completely different. It's a different ratio. So when they did their chi-square analysis here, they got a chi-square value of 44.22. And if we look at our chi-square distribution table, this value is greater than 7.85, which means that there is a statistical difference, right? So they got a value of 44.22. And they were expecting to see a value, that was not different, right? Somewhere below here, somewhere below 0.05. And they actually ended up with a value that was humongous, right? So there is a statistical difference. There is a difference, unlike what Mendel saw. So that they were perplexed. So then they did it again, right? They did the experiment again, and they ran the experiment in 1917 again, and they added plants. They added more plants thinking that, wait a minute, there's something wrong here, and they ended up with a chi-square value that was even greater, reassuring them that there was a statistical difference. So again, if we look at the chi-square value here, right, they got a humongous value of 973. So there was a statistically significant difference between what Bateson et al. Observed than what they expected according to Mendel's ratio. They were like scratching their heads, and they were like, well, what's going on? Now remember that now we know that if there is a statistical significant difference, we know that the genes are linked. If we have a value that's greater than the critical value. If it's less, we know that the genes are not linked. So they wondered, and they were like, well, what's going on? Did Mendel do the experiment incorrectly? Right? Because look at what we got. And they weren't able to answer that question, right? They had no idea. And this is what they wrote in their paper. The F2 results are complex and cannot be fully understood. These effects may be due to failure or segregation. The process which forms the different gametes is liable to disturbance. There is some coupling of pollen shape and color, so they couldn't explain it. But it wasn't until in 1911 Thomas Hunt Morgan established the fly room at Columbia University, and he studied linked genes. And again, this was over a period of 17 years, right? Thomas Hunt Morgan later won the Nobel Prize for Medicine in his work, 1933, and he established the chromosomal theory of inheritance. And I know that it might sound weird to us, right? Because we're like, yeah, we know of chromosomes, but Mendel didn't know of chromosomes, right? And even Bateson didn't know of chromosomes. So when Thomas Hunt Morgan published his experiments, right, he understood that some alleles and some genes could be linked, right? They could be on the same chromosome, unlike the genes that are unlinked. Okay? So if they're unlinked, they happen to be on different chromosomes. So what does that mean? Okay, that gave an explanation to Bateson's experiment, that what they were looking at in their experiments from 1904 to 1917, when they were looking at this type of sweet pea plant, they were looking at genes that were linked. Okay, so it doesn't mean that Mendel did wrong, right? Mendel was looking at unlinked genes and independent assortment. These guys were looking at genes that were linked, found in the same chromosome, some form of recombination, but most of the offsprings were like the parents. Okay? So this helps us understand the value of a chi-square analysis table, right? Especially in genetics. When you're running a genetics problem and you are trying to see if what you observe experimentally matches what is expected. Okay, so that's the importance of a chi-square table. I hope you guys are able to complete your own chi-square table, and that is it for today.